And these studies have to do with the role of ER stress response in neurodegeneration associated to Huntington's disease. That is the main topic in our lab, but also in epilepsy. And in case you are not familiar with the ER stress response, I give you first an introductory overview. And let me start by reminding you of the functions of the ER. And as you know, an important function is the synthesis of the transmembrane and secretory proteins, what is called the secretory pathway. And for that, the ER needs chaperones to follow the proteins that are synthesized at the time, and also enzymes involving glycosylation, and also the right redox balance for the formation of the disulfide bonds. Another function of the ER is the synthesis of sterols and phospholipids and the generation of biological membranes, and it is also the main storage of intracellular calcium. And whenever the cell faces circumstances when the ER has difficulties to perform these functions, then it will take place a phenomenon that is called an endoplasmic reticulum stress response, or also called unfolded protein response, because it's very often caused by the accumulation of unfolded proteins in the lumen that are sensed by 3 different sensors in the membrane of the ER that will activate 3 signaling cascades that will end up with the production of transcription factors like ATF4, cleaved ATF6, and secret splice XBP1 that will have then the opportunity to reach the nucleus and activate a program of transcription of different genes that will somehow help the ER to recover from this stressful condition. And the classic view of how is the ER stress response executed, it proposes that there are different levels of attempts of the ER to recover. The first attempt will come from the activation of PERK that will phosphorylate EIF2 alpha, and this will result in a global attenuation of translation of the messenger RNAs in the cell, so that it will decrease the load of proteins into the ER and help the ER to recover. The second layer of activity to help the ER to protect itself comes from the transcriptional activity of these transcription factors that I mentioned that will increase the production of ER chaperones like BIP, and also of components of the ERA, the endoplasmic reticulum associated protein degradation. These are a group of proteins that will take misfolded proteins in the ER, translocated outside, and immediately ubiquitinated for efficient degradation by the proteasome, and that way help the ER to recover. And finally, if the stress persists, then it is classically proposed that there is an induction of pro-apoptotic proteins like the transcription factor CHOP, the kinase J and K, and the caspase 12 itself that is associated to the ER membrane. So as I say, this is the classic canonical view of the ER stress response that is very well studied in yeast and cell lines, and it's also taking place in secretory cells when they have a high demand of production of the protein that has to be secreted. But I would like to draw your attention that there are also physiological situations in which there are partial or even total activation of these pathways, and that has been evidence from studying the different knockout mice lacking the different components of these signaling pathways. And for instance, it is well known that ATF4 participates in LTP without the need of any ER stress response execution because of any problem in the ER. I also would like to call your attention to the to the fact that there are other members of the ER stress, like for instance the transcription factor ATF5 that shares with ATF4 the sequence, the 5' regulatory sequences in the mRNA that allow to bypass the global attenuation of translation and selective translation upon these conditions of ER stress. So both ATF4 and ATF5 are selectively translated in these situations of ER stress, and ATF5 will also get to the nucleus and increase the transcription of chaperones and also of some anti-apoptotic genes like MCL1. And there are also some other proteins like the full-length ATF6 that are proteolytically cleaved by S1P and S2P substrates through the Golgi to finally decleave transcription factor, like the different CREB3, CREB4, and CREB3-like proteins, like CREB3-like2, that when reaching the nucleus will also increase transcriptionally ATF5. As you can see, I'm paying a lot of attention, bringing your attention to ATF5 because it's going to be one of the main proteins in the studies that I'm going to present afterwards. And now, what about the evidence linking ER stress response and disease? So there is clear evidence that there is a robust ER stress induction in different diseases, particularly inflammatory diseases, and also in metabolic diseases in which there are secretory cells involved, like for instance diabetes, and also in cancer. But what about neurodegenerative diseases? You know, in neurodegenerative diseases there are aggregation-prone proteins, and in the case of beta amyloid from APP or the prion protein that are produced through the secretory pathway, there is a clear, obvious link on how this aggregation of these proteins might be interfering with the function of the ER. It is not so intuitive in the case of proteins like mutant Huntington that aggregate in the cytosol or in the nucleus. But these proteins will be interfering or challenging with the other quality control systems in the cells, like the ubiquitin proteasome system and the autophagy system, and you know there is a crosstalk among them, and obviously with the ERAD. So here we have a possible connection on how aggregation of a cytoplasmic protein can lead to ER stress, but there is also plenty of evidence in most neurodegenerative diseases that calcium or redox disbalance takes place, and this might account also for a dysfunction of the ER. And more particularly in the case of Huntington's disease, there is a clear ER stress response execution in yeast and mammalian cell models, and there are also several examples of induction in mouse models and in postmortem human brain tissue, but there are also other studies that suggest a deficiency or maybe a disbalance among the different branches of the ER stress response signaling in HD human brain and mouse models. In fact, in our group, we reported a couple years ago that in the R61 mouse model that expresses N-terminal Huntington with a very long repeat, there are some markers of the ER stress response that are moderately increased or they are not clearly increased, but definitely there is one that is the production of cleaved ATF6 that is totally hampered, and in fact the levels are practically undetected in comparison with the normal levels in a wild-type mouse, and there is a concomitant increase in the full-length ATF6 form, so suggesting that the processing through S1P and S2P and through the Golgi to produce the soluble form of ATF6 is somehow hampered in Huntington's disease, at least in the R6 mice, and this accumulation of the full length gives in the R6 mice a dot-like pattern that is compatible with the retention in the ER, and also in postmortem human brain we found an accumulation of full-length ATF6, and also the nuclear accumulation that is found in age-control subjects is not seen in age-matched HD patients, therefore suggesting that ATF6 processing is impaired. So this would be suggesting that at least one branch of the ER stress is not well executed in Huntington's disease. So we see no processing of ATF6, not much change in ATF4, And then we wonder what would be happening with ATF5, that is an additional player similar to ATF4 in its regulation. And the first result we got, it was really remarkable. There was a very clear accumulation of ATF5 in the inclusion bodies in the neurons of the R6 mice, and this really prompted us to explore what might be the role of ATF5 in modulating ER stress response in Huntington's disease. But as soon as we started to get familiar with ATF5 literature, we got the surprise that ATF5 was supposed not to be expressed in adult brain. And that is nicely summarized in this cartoon from this review from Lloyd Green, that summarizes 5 or 6 papers that they clearly show that in progenitor neuroprogenitor stem cells there is a high level of ATF5. That's what is depicted with the red nucleus, and that for these cells to differentiate in vitro into mature lineages, neurons, astrocytes, these precursor cells, they have to decrease their levels of ATF5. If you keep them artificially high, they will never differentiate. And there is a nice correlation also in embryonic tissue that there is a decrease in the process of the differentiation of these precursor cells. And also, there are some papers showing that when there is a transformation of astrocytes and there is a malignant gliomas, there is a very high level of ATF5. But in view of these 5 or 6 papers, everyone assumed that ATF5 was not going to be expressed in normal adult brain, but we realized that actually no one had really mapped or analyzed the level of expression of ATF5 in normal adult brain. And because of this clear nuclear pattern neuronal pattern in the R61 mice, and also there was an experiment in the Allen Brain Atlas with a probe for ATF5 yielding a very clear neuronal pattern. So we decided then to first map ATF5 expression in adult mouse brain, and then to explore whether it is induced as part of the ER stress in primary neurons to get some functional clues on the role of ATF5, and finally to validate in vivo in a mouse model with a canonical induction of the UPR, that is the temporal lobe epilepsy mouse models that is generated by our collaborators Tobias Engel and David Henschel in Dublin. So first thing we did was to map with different antibodies the expression of ATF5, and we started in embryonic tissue, and we confirmed that in fact highest levels are found in neuroprogenitor areas, but also expression was not negligible in double cortin positive neuronally committed cells, and even early postnatal, this signal is colocalizing with NUN. And in the adult, it's very clear that immunohistochemistry gives you a neuronal pattern in the many different structures across the adult brain. And we also performed, with the help of Raquel Ibáñez and Pep Canals in Barcelona, in situ hybridization, and the neuronal pattern that was suggested by the experiment in the Allen Atlas was reproduced, and more importantly, double staining with immunohistochemistry for the neuronal marker shows that expression does take place in neurons, and on the contrary, there is no colocalization with the astrocytic marker GFAP. So this is demonstrating expression of the transcript in adult neurons, but not in astrocytes, in normal astrocytes. And we then moved to an injection of tunicamycin, that is a drug that interferes with the glycosylation machinery and induces a robust ER stress response. And when tunicamycin is injected into the striatum of a wild-type mouse, there is a clear increase of the signal of ATF5 in neuronal cells, and both in the striatum and in the cortex along the trajectory of the needle. So we then decided to move to primary neuronal cultures to verify the specificity of the employed antibodies and to get some mechanistic clues. And as you can see here, the primary neurons showed nice staining with ATF5, and the interfering RNA for ATF5 decreased ATF5 signal in the transfected cells, and also you could see the decrease by Western blot in nucleoaffected cultures. And when we treated the primary cultures with tunicamycin, we get a nice induction of all the canonical markers of the ER stress, and also an induction of ATF5. And this induction seems to be neuroprotective, because when we co-administer the interfering RNA for ATF5, we boost the neurodegeneration induced by tunicamycin, but if we overexpress ATF5, then we diminish the rate of neuronal death induced by tunicamycin. And then we decided to move to the animal model of this human disease with a clear neurodegenerative process that is that occurs with a clear paradigmatic UPR response. And this mouse model consists on kainic acid injection in the amygdala that through several synapses leads to a dramatic increase in glutamate release in the hippocampus, and it induces a status epilepticus with recurrent seizures along the life of the mouse, and there is a selective degeneration of the CA3 field with the CA1 and dentate gyrate fields being preserved in this model of focal seizure. And what Tobias Engel found is upon the injection of kainic acid, it was apart from the classic markers induction. By the way, ATF4 is increased in the degenerating area CA3. However, ATF5 was also increased, but that increase took place in the non-degenerating area CA1 and dentate gyrus, thus fitting with the protective effect that we have seen in the primary neuronal cultures. And finally, some pharmacological treatments that attenuate the neurodegeneration observed in the CA3 region of this animal model, like salubrinol, also increase ATF5 levels. So altogether, these data are demonstrating that adult neurons do express ATF5, that the ATF5 levels increase in neurons as part of the ER stress response, and this really seems to have a protective effect, or at least an anti-apoptotic effect. And I would like to mention another study that was performed in parallel, mainly by our collaborators in Ireland, and we found that the levels of a classically pro-apoptotic protein, CHOP, was increased too also in this mouse, in model of epilepsy-associated neurodegeneration. And these CHOP inductions occur in the three fields, in the one that degenerates, but also in those fields that do not degenerate. And interestingly, some manipulations that are neuroprotective in this paradigm, like the seizure preconditioning or the administration of salubrinol, as I mentioned before, that decrease the degeneration in this mouse model of epilepsy, also increase the levels of CHOP in the different regions of the hippocampus. And then we analyze what happened when you induce this epilepsy model in CHOP knockout mice, and we found that these mice lacking this supposedly anti-apoptotic protein, they have more, they are more vulnerable to the neurodegeneration induced by kainic acid injection, not only in CA3, but now even the CA1 and the dentate gyrus also exhibit neurodegeneration. This is not shown in this slide. And then we wonder what might be the mechanism behind this, and for that we perform a gene chip analysis to see which genes were differentially expressed when in mice subjected to status epilepticus lacking CHOP. And we found that the p53 target genes were upregulated in the CHOP knockout mice in response to the status epilepticus. And in fact, the p53 protein was increased itself, but not the p53 mRNA, therefore suggesting that the stability of the protein what is regulated, and that led us to focus on, to pay attention to the E3 ligase that controls the stability of the degradation of p53, that is MDM2. And we realized that in the promoter of MDM2 there are several responsive elements to which CHOP binds and might affect its transcription, and in fact MDM2 transcription is decreased and the protein is also decreased in the CHOP knockout mice. So there are more experiments in the paper that I don't have the time to go through them, showing that really p53 is responsible for that neurodegeneration. But let me just summarize the conclusions of this study. That is that in this ER stress response paradigm, there is an increase of CHOP in CA that in CA3 might be leading to neurodegeneration, but somehow by transcriptionally controlling MDM2, it is preventing p53 to induce even more apoptosis in other parts of the hippocampus. And in the CHOP knockout mice, this inhibitory loop is not present, and then there is a dramatic increase in p53 that is responsible for this increase in cell death. So then we can conclude that CHOP induction in vivo may also play a neuroprotective role in certain conditions, like for instance this temporal lobe epilepsy mouse model, and that it does so through transcriptional regulation of MDM2 and controlling the levels of p53. So as a general overview of the data that I presented, I just would like to remind you that we have found that ATF5 is expressed in neurons, and that it's induced as part of the ER stress, and probably it plays an anti-apoptotic role, that the ATF6 signaling cascade is impaired in Huntington's disease, and maybe this balance of one branches towards the others might have something to do to the pathophysiology of the disease. And also that to remind you that in some in vivo situations you may find that something that is very well established in cell models, like the anti-apoptotic, the pro-apoptotic role of CHOP, may be not so true in vivo situation like the model that I just presented. And finally, I would like to acknowledge the people that did the study. Jesus Torres Peraza did all the ATF5 study, and Charo Fernandez the ATF6 study, and Tobias Engel, a former student in the lab, but now postdoc or assistant researcher actually in Dublin, is the one that performed all the animals with the temporal lobe epilepsy model and the CHOP studies. And I would like also to acknowledge the other collaborators, and thank you for your attention. Is there any question? Yeah, Manuel over there. Steve.
All right, I'll take one first. Jose, it's really interesting that you found ATF5 even at baseline in neurons, and I just wondered, do you think that reflects some baseline level of ER stress in neurons, or do you think it plays some normal physiological function that we don't understand?
Well, there's no need for a physiological ER stress, because in fact there are two ATF5 mRNA isoforms, the so-called alpha and beta mRNA isoforms, and they are generated through alternative splicing, and only the beta isoform is subjected to this selective translation upon ER stress. The alpha isoform is, at least in other cell types and in other tissues, is well characterized to give a baseline level in normal conditions in the tissue. So it might be there just because of the non-inducible isoform of the mRNA. So Manuel?
Yeah, thanks very much. It was a great job. As far as I understood, it seems that the induction of this, say, non-canonical or more specific ATF5 pathway compared to the more canonical, the more general ATF4 pathway is somewhat exclusive.
Is it more?
Exclusive. I mean, you mentioned that you found ATF5 positive neurons in some areas and ATF4 positive neurons in other areas. So could you speculate about what can be the context specifics for this maybe particular induction of ATF5 in some areas of the brain and ATF4 in others?
Well, it might have to do with the stability of the protein once it is translated. Maybe it is more stable in some tissues than in other, and then their half-life is going to determine which one plays a more predominant role in one tissue and which one in the other tissues. But a very similar anti-apoptotic activity of ATF5 has been established in many different cell lines and other tissues, but no one had really paid attention to the brain because there was this notion that ATF5 was not expressed there.
Right. And because ATF4 can be either cytotoxic or cytoprotective, could be the relative contribution of ATF5 and ATF4 kind of balance this neuroprotection versus neurodegeneration?
Yeah,
role of ER stress.
Yeah, it might be the case. In the case of ATF4, I'm not really. I haven't really get in depth, but in the literature you can see downstream of ATF4 some transcription factors or just some genes that some of them you would assume that they will have a more pro-apoptotic, more anti-apoptotic. So maybe depending on the tissue, you will get a combination of gene expression that is more pro-apoptotic in one condition or less.
Thank you.
That's probably the explanation. Okay. Oh, okay. There's another question there.
Very nice talk. I'd like to know, I don't know whether you presented the data, but I didn't realize. Did you see as well an accumulation of HSP27 in the Huntington disease brain or in your mice models?
Well, that's unfortunately, Jesus, you heard that we got a Cohen project and also collaborative project very recently, but Jesus Torres, that is the postdoc that really did all this work, and that he was doing great in terms of publications, but he's quit science because he's now working as a clinician and MD in Palma de Mallorca, and that's part of the studies that he was conducting and is ongoing in the lab, but actually is orphan project in the lab at this moment. But if I remember, I think he has analyzed the canonical MCL1 and HSP27, but I don't have those data in my mind to be able to share those with you.